Triphala, current applications and new perspectives on the treatment of functional gastrointestinal disorders. This is printed July 18, 2018 in the National Library of Medicine, National Institute of Health. Abstract background. Ayurvedic medicine is based on natural healing methods that use herbal medicine to cleanse the body of toxins and to attain physical and mental regeneration. Triphala is one of the most important Ayurvedic supplements and is believed to have a beneficial effect on the entire gastrointestinal tract. Purpose. We aim to summarize available literature focused on the components of TLP and discuss their effectiveness and therapeutic value for improving lower GI symptoms of functional GI disorders, particularly irritable bowel syndrome, IBS, methods. The study is based on pertinent papers that were retrieved by a selective search using relevant keywords in PubMed and ScienceDirect databases. Results. The components of TLP are believed to cause restoration of the epithelium lining of the digestive tract, and by exhibiting mild laxative properties, facilitate passage of stool in the colon. TLP is rich in polyphenols, vitamin C, and flavonoids, which provide antioxidant and anti-inflammatory effects. It also contains various types of acids such as gallic, tubulinic, and additionally possess cytoprotective and antifungal properties. Triphala holds potential in improving lower GI symptoms and may be a valuable and effective addition to standard treatment of IBS. Supplementation of TLP herbal formulas alone or along with other probiotics can be recommended in ongoing clinical studies. Background. The pathogenesis of primary intestinal motility disorders is believed to be a multifactorial but neither biochemical nor structural abnormality has been displayed regularly other than in some forms of intestinal pseudo-obstruction. Patients with mild to moderate gastrointestinal GI motility disturbances often exhibit peripheral-generated symptoms with gut-based characteristics. Example: intermittent crampy abdominal pain, indigestion, nausea, bloating or feeling of incomplete evacuation, whereas others experience noxious, persistent, and severe symptoms with marked GI tract function impairment, usually accompanied by somatic psychiatric comorbidities or extra-intestinal symptoms, such as headache, musculoskeletal pain, dizziness, or fatigue. Irritable bowel syndrome, IBS, is the most common type of functional GI disorders and is characterized by recurrent abdominal pain and disordered bowel habits. IBS patients represent almost 50% of all individuals who seek medical assistance with primary care physicians or GI specialists. There is no specified marker for IBS, and its diagnosis is symptom-based. The cause of IBS development remains undefined, which makes it difficult to diagnose and treat. Recent studies highlight the interaction between luminal variables, example foods and microbes dwelling in the digestive system, the epithelial barrier, and the mucosal immune system, as a complex network involved in the pathogenesis of IBS. The overstimulation of the mucosal immune system prompts the infiltration of mucosal immunocytes, particularly mast cells, and promotes secretion of various mediators, such as proteases, histamine, and prostanoids, which further propagate permeability dysfunction and activate atypical neural reactions involved in abdominal pain perception and gut motility. With respect to a wide variety of symptoms seen in IBS, patients tend to use different methods for symptom relief, including alternative treatments such as acupuncture or dietary supplements. Usually, changing nutritional regimen and lifestyle are the first steps taken by patients, mainly because such changes can be incorporated into daily life from one day to another without an in-depth clinician's consultation. An extensive research has provided convincing evidence for beneficial effects of nutritional supplements, including the intake of probiotics, prebiotics, and symbiotics, and herbal medicine products on the gut microenvironment. Herbal medicines have been widely used for the treatment of IBS patients. Probiotics and dietary supplements can normalize bowel movements, reduce visceral hypersensitivity, and by stimulating the immune system, reduce inflammation and gut permeability. Alternative treatments still warrant scientific validation of their efficacy. Phytochemical constituents of Triphala. Triphala is an Ayurvedic herbal formulation of dried fruits from 3 herbal plants in equal proportions. Terminalia chabella, the black myrobalan. Terminalia barylica, the bastard myrobalan. And Plantaeus embellica, the emblic myrobalan or Indian gooseberry. Fruits of T. chabella that are harvested in the spring are a rich source of tannins. Example, chabullic acid, chabulinic acid, neochabulinic acid, coralligin, chabellic acid, gallic acid, ellagallic acid, pentogolan, terchamin, and terflavin. T. chabella fruits additionally contain flavonoids, the luteinins, rutins, and quercetin, and other phytochemicals, anthraquinones, saponins, bd-glucogalin, 1,3,6-triglycose, and 1,2,3,4,6-pentaogali, starches, amino acids, aspartic acid, glutamic acid, arginine, proline, and lysine, fructose, succinic acid, B-citosterol, and fatty acids. So far, T. chabella fruits have been utilized in conventional pharmaceuticals to battle ailments of the upper respiratory tract, the GI tract, urinary tract, and skin. Antimicrobial activity. Triphala is known to exert antibacterial action against a variety of gram-positive and gram-negative bacteria. Example, Streptococcus mutans, a gram-positive anaerobic bacteria mostly found between adjacent teeth or in the deep crevices on occlusal of teeth, or Hypobacterial pylori, the principal cause of inflammation of the gastric mucosa. Worth mentioning due to impaired immune system and this ability to eliminate bacteria from the gastric mucosa, the bacteria-generated symptoms reach beyond the stomach and very often include abdominal pain, heartburn, belching, and/or flatulence. The bacteria also affect lower GI motility and consequently result in constipation. Triphala is also an antiviral operator that acts against swine flu and other viruses, HSV1, HIV1, and cytomegalovirus or hepatitis B virus, HBV surface antigen. Antioxidant activity. Since antioxidants play an important role in protecting the body against damages caused by reactive oxygen species, ROS, many studies have been devoted to uncovering the antioxidant potential of TLP. TLP possesses superoxide radical scavenging and hydroxyl radical scavenging activity as shown in vitro and in vivo studies. Worth mentioning, the aqueous and alcoholic extracts of the individual constituents enhance the activation of macrophages due to their ability to scavenge free radicals and neutralize ROS. The alcoholic concentrate of T. Triphala and T. bellerica extracts induce the production of ROS in macrophages, mainly through the prophagocytic activity of gallic acid. Similar results were reported in studies with P. embolica, the amla fruit extract. Of note, amla is rich in antioxidants, flavonoids, carotenoids, and vitamin C. In fact, it is considered as one of the richest sources of vitamin C, as proved in both in vitro and in vivo studies. The antioxidant effects of amla was assessed in rodents by measuring the activity of oxidative free radical scavenging enzymes, including the superoxide dismutase, SOD, catalase, CAT, glutathione peroxidase, GPX, as well as lipid peroxidation. In line, animals were allocated to one of the intervention groups to receive either an active compound of amla or diprenyl, a monoamine oxidase B inhibitor, used as a positive control. Amla and diprenyl increased the activity of SOD, CAT, and GPX, and suppressed lipid peroxidation after 7 days of treatment, which confirmed the antioxidant efficacy of amla, attributed to the presence of P. embolica, an active compound of tannoids. In addition, phytochemicals found in P. embolica were also considered as good metal ion chelators, as they hamper the oxidative cascades. Hypolipidemic effect. A study by Manjunatha and others showed that volunteers given amla for 16 weeks had significantly improved HDL-LDL ratio and glucose tolerance, and reduced cholesterol level in comparison to the control group receiving 500 milligrams a day of vitamin C. The anti-atherosclerotic effects of P. embolica was also evaluated in rabbits fed with cholesterol-rich diet. After 60 days of supplementation with amla, the serum cholesterol, triglyceride, phospholipid, and LDL levels were reduced by 82, 66, 77, and 90% respectively. Importantly, the aortic plaques in treated animals were regressed, suggesting a potent hypolipidemic action of P. embolica. Another study revealed that flavonoids extracted from amla exhibited intense hypolipidemic and hypoglycemic activities, and increased the level of hemoglobin in rats. Apparently, TLP is known to be one of the best Ayurvedic medicines with antioxidant and anti-aging properties. Anti-cancer activity. The major phytoconstituent of TLP, gallic acid, is believed to have a great impact on the anti-cancer properties of the polyherbal formulation of Ayurveda, as it inhibits cancer cell proliferation. Hence, it may be considered as a major determinant of anti-mutagenic and cytotoxic activity of TLP in human colon cancer cells and stem cells. The methylenic extracts of TLP exerted a dose-dependent anti-proliferative properties and pro-apoptotic action independently of the p53 status of the cells. Moreover, treatment with TLP extract induced apoptosis via elevation of BAC's BC1-2 ratio, and suppress the CMRC, serine D1 expression, and therefore decrease cell proliferation and colony formation. Recently, Gu and others showed that one of the constituents of TLP, the P. umbilica, delays mitotic progress, induces and accelerates apoptosis, and increases genomic instability of human adrenocarcinoma cell line in a dose-dependent manner and in human colon epithelial cells. These results imply that P. umbilica may protect human colon epithelial cells from genetic and mitotic changes, and simultaneously indicate the necessity to evaluate its extract in depth in animal studies. The tumorigenic effects of TLP and its components were also determined in other cancer cell lines and in vivo studies. P. umbilica and a preparation containing 50% of its natural extract given to mice significantly reduced ascites and Dalton's lymphoma. Ascite cells induce solid tumors by inhibiting the cell cycle, regulating enzymes such as CDC25 phosphatase. TRP also hampered benzopyrene-induced forced tumorigenesis, suppressed the growth of MCF7 breast cancer cells, protected against radiation-induced oxidative damages, and inhibited the proliferation of CAPN2 and PXPC3 human pancreatic cancer cells through the activation of ERK, P53, and capsase-3 cascade. The extract of TLP and its components hold potential as adjuvant therapy to the currently used anti-cancer agents. However, currently available data warrant more extensive preclinical and clinical investigations. Immunomodulatory activity. It's been observed that through the range of biologically active compounds, gallic acid, lactic acid, gibberellinic acid, TLP can help reduce inflammation by lowering the expression of pro-inflammatory mediators. The alcohol extract of the dry, ripe fruit of T. Triphala protected cells from oxidative stress by inhibiting lipid peroxidation, increasing the levels of glutathione, superoxidase dimutase, and CAT, and by changing the mRNA expression of selected pro-inflammatory cytokines. TLP also improved the natural killer cell viability and antibody-dependent cellular cytotoxicity in Dalton's lymphoma-bearing mice. The immunomodulatory characteristics of TLP have also been tested using carbon clearance test and delayed type hypersensitivity response in vivo. TLP mega extract, when administered orally at low and high doses of 100 and 500 milligram kilogram respectively, increased the carbon clearance index, which is reflected in improved phagocytic function of mononuclear macrophage and nonspecific immunity. Moreover, the extract stimulated the production and release of T cells. Many studies showed that modulation of NF-beta activity shows a promising target for the treatment of inflammation-related disorders, and since TLP can act via this pathway, it makes it a suitable option for the prevention and management of many diseases. In line, the study showed that gallic acid, seed, and TLP reduce the formation and release of pro-inflammatory mediators by decreasing DNA binding in the NF-beta in cells treated with lipopolysaccharides. In addition, the components of TLP and their metabolites were able to suppress the expression and production of the TNF-alpha, IL-1 1-beta, IL-6, COX-2, and NO in raw RAW-TS2647 cells, and primary macrophages stimulated with LPS through the inhibition of redox-sensitive kinase activity and blocking the NF-beta-nuclear translocation. The mixture of cibulonic, gallic, and oleic acid exerted immunomodulatory activity, and thus makes the Triphala a potent plant material used to prevent cancer development. TLP and its extracts have been useful in mimicking the action of neutrophils by preventing elevation of several cytokines. During the inflammatory stress and immunosuppressive activity of TLP is mainly linked to its inhibitory action on mitogen-induced T lymphocyte proliferation of humoral and cell-mediated immunity. Taken together, these observations indicate the immunosuppressive effect of TLP and its extracts is attributed to decreased production of inflammatory mediators via the NF-beta pathway in experimental induced inflammation, and therefore may provide an alternative approach for the treatment of inflammatory and autoimmune diseases. Is TLP an effective remedy for improving GI symptoms? Literature data does not provide any clear evidence that shows the effect of TLP on the human digestive tract. Available studies indicate bidirectional activity of TLP, very often focusing on a particular component of TLP, as it is likely to either accelerate or slow down intestinal peristalsis. Mohmand and others evaluated the effects of P. umbilica in constipation and indigestion in vivo and showed that P. umbilica possessed prokinetic and laxative activities and exerted spasmodic effect in the isolated ileum and jejunum of guinea pig and rabbit, respectively. The stimulatory action on the gut was supposedly mediated through the activation of muscarinic receptors. Another study was designed to evaluate the enteroprotective effects of TLP formation and methotrexate-induced damages in rats. Different formulations of TLP solutions protected from intestinal injuries by improving epithelial cell integrity of tight junctions and restoring the brush border membrane of intestine, and decreasing the myeloperoxidase and xanthine oxidase levels in the gut mucosa. The observed effect has been attributed to flavonoids, oleic and gallic acids in TLP formulation. Depending on the proportion of the T. cibula, T. bellerica, and P. umbilica, the level of the injuries varied. It was suggested that unequal formulation of the ingredients in TLP example the highest proportion of P. umbilica rather than its equal formulation provided better protection of intestinal microvilli. Possible clinical application of TLP has also been assessed in patients with functional constipation. An open-label, prospective, interventional, and exploratory clinical trial conducted by Munshi and others demonstrated that 1-week treatments of TLP increases the average weekly bowel frequency by 64.4% and by 79.5% after 2 weeks. The increase in the mean bowel movements maintained 7 days after observatory period was approximately 18% higher than that of baseline value. Another study showed that one of the constituents of TLP, T. tubuli, ameliorated digestion and severe continuous constipation, as well as reduced intestinal cramps. It has also been shown that the long-term 45-day treatment with TLP improved the number, frequency, and consistency of stools excreted per day, and decrease the abdominal pain and bloating in healthy patients versus control in individuals who did not receive TLP. No side effects in the treated group were reported. It seems that TLP can be endorsed in the primary line treatment for patients with delayed GI motility. However, several other studies indicated TLP as an anti-diarrheal agent, thus disproving an earlier stated assumption. The anti-diarrheal effect of water and alcoholic solutions of TLP were examined in a rat model of castor oil-induced diarrhea. Each solution, independently of the dosage used in the study, prolonged the whole GI transit and increased the weight of feces. In oral toxicity testing, aqueous and alcoholic solutions of TLP were viewed as effective and safe in the dosage up to 1,750 milligrams per kilogram. It has been reported that the presence of phytochemicals in TLP, such as alkaloids, flavonoids, tannins, terpenes, sesquiterpenes, could be responsible for the antidiarrheal, anti-secretory, and spasmolytic effects of TLP. The gut microbiota can protect the host from invading pathogens, maintain immune function, and help absorb dietary nutrients. Quantitative and/or qualitative changes in the composition of intestinal microbiota can modulate the intestinal barrier function, particularly via the release of short-chain fatty acids, and thus induce systemic immune response. There is a correlation between the abundance of specific microbial populations and the occurrence of GI symptoms. An increase in bifidobacterium is thought to be negatively correlated with intestinal symptoms such as bloating. Certain herbal extracts have proven its effectiveness in clinical trials in patients with FGID. Therefore, some attempts have already been made to evaluate the bioactivity of TLP and its extracts. Due to the presence of P. emblica, TLP inhibits the growth of pathogenic microorganisms in the intestines, which may aggravate intestinal symptoms. Polyphenols in TLP can modulate the human gut microbiota by promoting the growth of beneficial bifidobacterium and lactobacillus species and inhibiting the growth of undesirable intestinal residents, such as E. coli, which may induce the inflammatory reaction. Moreover, TLP-derived polyphenols can be transformed by the human intestinal microbiota into various active metabolites, including urolithrins, which modulate the inflammatory process by generating anti-inflammatory compounds and preventing from oxidative injuries of enterocytes. The possible manipulation of gut microbiota by TLP extracts may represent a new strategy in the development of functional foods or nutraceuticals and become an effective way of restoring gut microbiome in patients with impaired lower GI function. Example, patients with IBS. Currently, there are no clinical trials assessing the effects of herbal formulations of TLP on clinical courses of IBS. Thus, it's difficult to determine the full value of TLP supplementation. Additional studies on human subjects are warranted given the recently revealed enteroprotective, prokinetic, and antibacterial properties of TLP in the GI tract. Concluding remarks. Medicinal plants play a significant role in primary health care in many developing countries and have attracted renewed interest in developed countries over the last decades. Since natural products constitute a key source of drugs in medicine, the interest around them is increasing, together with the number of studies trying to reveal their biological effects on humans. Since TLP is a multi-ingredient formulation, it's likely that it could prove its effectiveness by modulating multiple targets rather than acting on a single one. Various compelling preclinical, in vitro, and in vivo studies substantiated antioxidant, antimicrobial, antimodulatory properties of TLP and its components, and proved its usefulness in an array of diseases, including hemorrhoid, skin disease, asthma, dysentery, uterine debility, anemia, diabetes, heart disease, and others. Although some attempts were made in order to determine the effects of TLP on lower GI tract, its action is still not well defined. Nevertheless, it seems that the herbal combination of TLP may bring benefits as adjunct to currently applied treatment among patients with FGIDs, including IBS.